Way back in 1937, a famous biochemist named Hans Krebs discovered a path-breaking biochemical cycle. Now, he faced a lot of hurdles along the way. Initially, when he came up with the idea or hypothesis, it was rejected flatly by his contemporary. Later on, when he formulated the entire cycle and decided to publish it in a reputed journal, the journal rejected his research paper, saying that they had lots of other research papers to be published and suggested him to publish the article somewhere else, which he did and was successful. Today, the biochemical cycle is quite popular and is considered to be the major source of energy production in most cells. I'm talking about citric acid cycle, which is the topic of this session. So let's have a look at what are the learning objectives of this session. At the end of the session, you should be able to define the cycle. You should know the site, the subcellular site, along with the key enzymes and the main reactions in the cycle. Obviously, you need to know the starting material and the end products, along with the energetics, how much of energy is produced during the entire cycle, and last but not the least, how the cycle is regulated. So we'll move on to the first part, that is the definition. So citric acid cycle is the oxidation of acetyl-CoA to form carbon dioxide and water. At the same time, a lot of energy is produced, which is utilized by cells. It is also called as the Krebs cycle, named after the person who discovered, the scientist who discovered the cycle, that is Hans Krebs. Another point I would like to draw here is Hans Krebs is also known for his work that involves urea cycle. So there are two cycles in which Krebs played a role in discovering and formulating. That is, one is citric acid cycle and the other one is urea cycle. Now, citric acid cycle is also called as a tricarboxylic acid cycle because two of the intermediates are tricarboxylic acids, that is citrate and isocitrate. These are the intermediates of TCA cycle or citric acid cycle. Now, what do you mean by a tricarboxylic acid? Tricarboxylic acid is one which contains 3 carboxylic groups in the structure. So now we'll move on to the significance. Now, citric acid cycle is considered to be the final common oxidative pathway for all 3 metabolic pathways, that is, which involves carbohydrates, fats, and amino acids. Now, you know, carbohydrates, fats, and amino acids are the fuels that are required for energy production. So I'll just show you how these 3 help form acetyl-CoA, which is the substrate that enters into TCA cycle. So let me write the 3 fuels that are required first. We have carbohydrates, and then we have fats and proteins. Now the major carb which is utilized by our body is glucose. Glucose within the cells usually undergoes glycolysis. Through a series of reactions, it forms pyruvate. Pyruvate undergoes an oxidative reaction, oxidative decarboxylation reaction, with the help of the enzyme pyruvate dehydrogenase to form acetyl-CoA, which is the starting material of TCA cycle. Okay, this is one way by which carbs are broken down to form acetyl-CoA. The other fuel, which is fats, is broken down to acetyl-CoA through a process called as beta oxidation. And last but not the least, we have proteins, which are made of amino acids. Now, certain amino acids called as ketogenic amino acids. Ketogenic amino acids are those which can be utilized for formation of ketone bodies. So, ketogenic amino acid, the common examples are that of leucine and lysine. Then you also have certain amino acids which are partly glucogenic and partly ketogenic. So partly glucogenic and ketogenic examples are phenylalanine, tyrosine and tryptophan. So these amino acids can lead to the formation of acetyl-CoA. So this is how all the three fuel molecules are utilized for the formation of acetyl-CoA, which then enters into the TCA cycle to produce energy, which is the whole idea of this session. So again, it generates energy, and along with that, there are certain intermediates that are formed that are utilized to form important products in the body. That I'll come to this a little while later. So the major site is basically, this cycle occurs in most of the cells except the cells which do not have mitochondria, because that is the subcellular site in which TCA or the enzymes of TCA cycles reside. So cells which do not have mitochondria is red blood cells, RBCs. Mature RBCs or erythrocytes are the ones which lack mitochondria. Hence, TCA will not operate in RBCs, whereas it will operate in all the other cells where mitochondria exist. And the starting material obviously is acetyl-CoA. Like I have just told you, here you have all the three fuels which are broken down to ultimately give you acetyl-CoA, and which enters acetyl-CoA in turn enters into TCA to give you energy. Citric acid cycle or the TCA cycle contains 8 steps catalyzed by 8 enzymes. So the first step, we have acetyl-CoA, which is a 2-carbon containing compound. It combines with one of the intermediates of the TCA cycle known as oxaloacetate. Oxaloacetate is a 4-carbon containing compound. Together, these two combine to give you the next intermediate, which is citrate, a 6-carbon containing compound. So now you can see there's a coenzyme A which is attached to the acetyl group. The coenzyme A is released out as active coenzyme A, and during the process a water molecule is required. This step is catalyzed by the enzyme citrate synthase. Citrate is the product form and synthase meaning synthesis. This is an irreversible step. In the next step, citrate undergoes dehydration. This is a reversible step where a water molecule is lost, or it undergoes dehydration to form an intermediate called as cis-aconitate. The enzyme which is required for this reaction or step is called as aconitase. Okay, so this is the first reaction and this is the second reaction. Now cis-aconitate undergoes another reversible reaction where water is added once again or hydration occurs. So again, this is a reversible reaction where water molecule is added to cis-aconitate, and the same enzyme which catalyzed the previous step will catalyze the next step as well. So we have aconitase again over here. And you get an isomeric form of citrate as a product which is known as isocitrate. Isocitrate is a 6-carbon containing product. Now, you have to remember that these two steps are actually one combined step where you have citrate getting converted to isocitrate. Now this dehydration and hydration which occurs here is done in order so that there is rearrangement of the atoms which are present in citrate so that the isomeric form is formed. So this entire step is considered as the second step in citric acid cycle, which is catalyzed by aconitase. Now, aconitase has more than just one function. Other than being an enzyme of the citric acid cycle, it plays a major role in protein regulation. So, it is said that it plays a major role in iron metabolism, especially where it regulates certain proteins involved in iron metabolism, such as transferrin receptors, and ferritin. All these three proteins are involved in iron metabolism, and aconitase plays a major role in regulating those proteins. So let's move on to the pathway as such. In the next step, isocitrate undergoes oxidative decarboxylation. This is a reversible step. Now, oxidative decarboxylation is a step where there's oxidation and decarboxylation occurring. Oxidation here is removal of hydrogen, and decarboxylation is nothing but removal of carbon dioxide. So the hydrogen acceptor is NAD+. NAD+ stands for nicotinamide adenine dinucleotide. So NAD+ accepts hydrogen from isocitrate and gets converted to NADH. And at the same time, there's a decarboxylation reaction occurring simultaneously where you see carbon dioxide is removed. Now, it is said that this NADH which is formed enters into the electron transport chain, which is present in the mitochondria itself, and produces some amount of energy. Now, how much energy is produced? I'll talk about that in a little while later. So, this is an oxidative decarboxylation reaction, a reversible reaction. And here you see that isocitrate gets converted to alpha ketoglutarate. With the removal of a carbon dioxide from isocitrate, the 6-carbon compound gets converted to a 5-carbon compound. The enzyme which is required to catalyze this step is known as isocitrate dehydrogenase. And this becomes the third step. Now, in the next step, what happens is alpha ketoglutarate gets converted to succinyl-CoA, which is the next intermediate formed. Again, this is an example of an oxidative decarboxylation reaction where you have NAD+ as the hydrogen acceptor. NAD+ accepts hydrogen from alpha ketoglutarate and forms NADH. Which eventually enters into the electron transport chain and produces energy. At the same time, you have carbon dioxide which is released. This reaction also requires certain other coenzymes or cofactors such as thymine pyrophosphate, lipoic acid, okay, these are all derivatives of vitamin B, and then you also need an active coenzyme A. The reaction is catalyzed by the enzyme alpha ketoglutarate dehydrogenase. As you can see, there is removal of hydrogen, so you call this enzyme dehydrogenase. This is the fourth step, and this is an irreversible step. In the previous step, you saw carbon dioxide was released. So you can see that the 5-carbon atom over here, 5-carbon compound over here, which is alpha ketoglutarate, loses a carbon dioxide to form a 4-carbon compound, which is succinyl-CoA. Now, succinyl-CoA is then converted to succinate, which is the next intermediate. During this reaction, succinyl-CoA is said to have a thioester linkage. So the energy which is stored in succinyl-CoA is transferred on to GDP, which is guanosine diphosphate, to form GTP directly. So energy is directly formed here in the form of guanosine triphosphate. And at the same time, you can see coenzyme here, over here, attached to succinyl-CoA. That gets released as free coenzyme A. So energy is released in the form of GTP, and coenzyme A is also released. This step is catalyzed by the enzyme succinate thiokinase. This is the fifth step. Now, if you noticed, there's a slight difference in the energy production between this step, that is the fifth step, and the energy which is produced at the fourth and the third step. Now, in the fourth and the third step, you get NADH, which is a reducing equivalent, which is formed here. And that reducing equivalent enters into the ETC cycle or electron transport chain, where it forms energy. So that is an example of an oxidative phosphorylation reaction where energy is produced from reducing equivalents within the electron transport chain. Whereas on the other hand, at the fifth step here, you can see energy is directly produced. There's no reducing equivalent form which enters into the electron transport chain and forms energy. Rather, whatever linkage, thioester linkage is present here in succinyl-CoA, it is directly transferred to GDP to form energy in the form of GTP. So this direct formation of energy from the substrate is called as substrate level phosphorylation. Whereas energy production where the reducing equivalents enter into the electron transport chain and produces energy is called as an oxidative phosphorylation. So this was the fifth step. Now we'll move on to the next. Succinate gets converted to fumarate. This is catalyzed by the enzyme succinate dehydrogenase. It's an oxidative reaction where hydrogen is removed, dehydrogenase. The hydrogen acceptor over here is FAD. FAD is flavin adenine dinucleotide. FAD accepts the hydrogen from succinate to form FADH2, which then enters into the ETC electron transport chain to produce energy. So again, the energy production, I'll talk about it a little while later. This is an example of an oxidative phosphorylation where the reducing equivalent, that is FADH2, enters into the electron transport chain and produces energy. Now, next, fumarate. Let me write here. This is again, succinate is a 4-carbon compound and fumarate again is a 4-carbon compound. Now, fumarate undergoes a hydration reaction where water is added to fumarate to form the next intermediate, which is called as malate, and the enzyme responsible is fumarase. This is the next enzyme. This becomes the 6th step and the fumarase step becomes the 7th step. Okay, so now we are done with the 7th step. In the next step, which is the final step, where malate gets converted to oxaloacetate, the enzyme required is malate dehydrogenase. This is an oxidative reaction where hydrogen is removed. A reversible step again. And the hydrogen acceptor is NAD+. NAD+ accepts the hydrogen from malate to form NADH, which again enters into the ETC, electron transport chain, to form energy. This becomes the 8th step. So you can see very well in the citric acid cycle, there are 8 steps and 8 enzymes, and the main thing over here is the production of energy, especially at steps 3, 4, 5, and you have even at 6 and 8. So now let's have a look at how energy is produced. Like I've said, you have reducing equivalents which are formed, which enters into the electron transport chain and produces energy. Now, as per the recent rules or as per the recent findings, it is said that when one molecule of NADH enters into the electron transport chain, it produces 2.5 ATP. Okay, this is the latest rule. Earlier it was thought that, one molecule of NADH, when it enters into the electron transport chain, it forms 3 ATP. Now it is no longer followed. Remember, the latest rule is one molecule of NADH produces 2.5 ATP, whereas one molecule of FADH2 produces 1.5 ATP. Earlier it was thought that one FADH2 produces 2 ATP. So now it's 1.5 ATP. So now we'll just calculate how much of ATP is synthesized or produced during the entire cycle. Now we'll see step number 3. Okay, you have NADH produced over here, enters into the electron transport chain and produces energy. As I've said, one molecule of NADH produces 2.5 ATP. So I'll write 2.5 over here. 2.5 ATP is produced here. Okay, this is one point. The next point is over here at step number 4, where again you have NADH produced, which enters into the ETC, and this produces again 2.5 ATP. Okay. Now in the next step, which is the 5th step, you have energy which is directly produced at substrate level phosphorylation. So we can assume that 1 GTP is nothing but 1 ATP. So I'll write 1 ATP here. This is the energy formed. Okay. And then we have FADH2, which enters into the ETC. And as I've said, one molecule of FADH2 produces 1.5 ATP within the electron transport chain. The last reducing equivalent is formed at the 8th step, where you have NADH produced, which enters into the ETC producing 2.5 ATP. Now, if you add up all the ATPs that are formed, you have 2.5 ATP here. Now you have 5 ATP till here when you add up the energy produced. 5 ATP plus 1 ATP, you have 6 ATP. 6 ATP plus 1.5, you get it as 7.5. And then finally you have 7.5 plus 2.5. The net energy production is 10 ATP during the conversion or during the oxidation of acetyl-CoA to carbon dioxide and water. So that is the net energy produced. Now, in addition to all these, you will also need to know what are the various inhibitors of the pathway. I'll just cover it up here itself, since I've already mentioned the entire cycle. I've written the entire cycle here. So there are 3 main inhibitors of TCA cycle. The first inhibitor, which is fluoroacetate, inhibits aconitase, which is this. Okay, it inhibits these 2 steps in the pathway, which is catalyzed by aconitase. The kind of inhibition that occurs here is non-competitive inhibition. The next inhibitor is known as arsenite. Again, arsenite inhibits the enzyme alpha-ketoglutarate dehydrogenase through non-competitive inhibition. So the method of inhibition is the same, non-competitive inhibition, in case of fluoroacetate as well as arsenite. And the last inhibitor for the pathway is here at the level of succinate dehydrogenase, where you have malonate, which is the last inhibitor. It inhibits the enzyme succinate dehydrogenase through competitive inhibition. So with this, I'm done with the inhibitors as well as the energetics along with the reaction of the pathway citric acid cycle. So moving on, before I tell you the same things, whatever I've just spoken in the previous slide, I forgot to mention one thing. I have a mnemonic for you all to remember at least the intermediates of the pathway. I know there was a lot of things in the previous slide, including what are the coenzymes, what cofactors are used in each step and all. So there's so much to learn and so much to understand from the cycle itself. At least you should be able to remember the intermediates of the cycle. So there's a mnemonic for that, to remember the intermediates. And I made this up. you can choose to, make up your own mnemonics. So the mnemonic that I follow is, can I add some salt for my omelette? Now, if you're a vegetarian, you can change this to anything. Since I'm a non-vegetarian, I've written omelette. So C stands for citrate. I stands for isocitrate. A stands for alpha ketoglutarate. S stands for succinyl-CoA. Then the other S stands for succinate. F stands for fumarate. And obviously M and L stands for malate and oxaloacetate. Now you have the confusion of two S's over here, so you'll wonder which one comes first. Is it succinyl-CoA or succinate? Remember, succinate is always late. So therefore, succinate will always come after succinyl-CoA. So this is one way you can remember all the intermediates of the pathway. Now moving on to inhibitors, I've already mentioned in the reaction in the previous slide how various inhibitors inhibit some of the enzymes in TCA cycle. So we have fluoroacetate, aconitase and arsenide, which inhibits alpha ketoglutarate. Both of these are examples of non-competitive inhibition. And the last one is malonate, which inhibits succinate dehydrogenase. This is an example of a competitive inhibition. Moving on to the next one, which is energetics of TCA cycle. Again, it was explained in the previous slide. I won't go into it. Remember, you have energy generated at various steps. So I've mentioned the enzymes over here. The total energy which is formed is 10 ATP. And this is 10 ATP is based on the new rule where one molecule of NADH produces 2.5 ATP and one molecule of FADH2 produces 1.5 ATP. So that was the energetics produced with one molecule of acetyl-CoA, which gets converted to carbon dioxide and water. Now, if you consider the complete oxidation of glucose, considering that glucose is the principal carbohydrate ingested and broken down in our body in various cells, if you consider the total energetics of glucose oxidation, glucose generally undergoes glycolysis. So during anaerobic oxidation, you have pyruvate formed. During aerobic oxidation, you have pyruvate formed. In the presence of oxygen, you have pyruvate formed. During aerobic condition, you have 7 ATP molecules generated, 7 molecules of ATP generated. Then pyruvate undergoes oxidative decarboxylation by the enzyme pyruvate dehydrogenase, which I mentioned in the next step here, to give you acetyl-CoA. During this step, conversion of pyruvate to acetyl-CoA, you have 5 ATP generated. And then finally the acetyl-CoA enters into TCA. And during the oxidation of acetyl-CoA within the TCA, you have 10 ATP generated. But have a look at this. I have mentioned 10 ATP into 2. Why? Remember, acetyl-CoA is a 3-carbon containing compound. But the initial substrate, which is glucose, which gives you acetyl-CoA. Glucose is a 6-carbon containing compound. Therefore, on complete oxidation of glucose, which is a 6-carbon containing compound, you get 2 molecules of acetyl-CoA. So 3-carbon acetyl-CoA plus 3-carbon acetyl-CoA is the product formed at the end of oxidation of glucose, which is why it is said that acetyl-CoA goes into the TCA cycle twice. So you get 20 ATP. When you add this all up, you get 32 ATP, which is the total energy formed during the aerobic oxidation of glucose. On the other hand, if you see under anaerobic conditions, I have mentioned 2 ATP is synthesized. Now generally, during glycolysis, when glucose is converted to pyruvate and then lactate, This happens only in anaerobic glycolysis, where glucose gets converted to lactate. The net energy production is not 7 ATP, but you get 2 ATP formed here in this step. This is in anaerobic glycolysis. The next reaction, which is pyruvate to acetyl-CoA and TCA cycles, do not occur because of the lack of mitochondria. So because mitochondria is not there in anaerobic condition does not occur within mitochondria, these two steps do not occur. So therefore, you do not have energy produced in these two steps. Directly, whatever energy is produced during anaerobic glycolysis, that is 2 ATP, is the total energy produced during oxidation of glucose. So this is it about energetics of glucose oxidation. Now coming to regulation, there are two main things that is kept into mind when you talk about regulation of TCA cycle. First is the availability of substrate. The substrates are required for TCA cycle to proceed. So you need all the fuel molecules such as carbohydrates, proteins and fat to be broken down to form acetyl-CoA, which enters into the TCA cycle. The next thing is the need of ATP for the cell. So if the energy of the cell is maintained, it is adequate, then TCA cycle won't operate. If the energy of the cell is low, then the citric acid cycle will operate so as to provide energy for the cell. And the regulation occurs mainly on allosteric regulation. So you can see these are the key enzymes that help regulate the TCA cycle. You have citrate synthase, which is the first enzyme catalyzing the first step. Then you have isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. So in case of citrate synthase, the presence of energy within the cell will inhibit the enzyme. Next, when you see isocitrate dehydrogenase, the presence of energy as well as the reducing equivalent form, because the reducing equivalent will also produce energy when it enters into TCA. These two will inhibit isocitrate dehydrogenase, whereas ADP will activate the enzyme. And the last but not the least is alpha ketoglutarate. The product formed, which is succinyl-CoA, is known to inhibit the enzyme. Also, presence of energy as well as the reducing equivalent as well will inhibit the enzyme. So this is the regulation of tricarboxylic acid cycle. Now, there are two important aspects of tricarboxylic acid cycle. First of all, energy production. The other one is formation of important substrates or important products from the TCA cycle. Now, TCA cycle is considered to be amphibolic. What do you mean by amphibolic? Amphibolic is nothing but a pathway or a cycle where both formation as well as breakdown occurs. So we have studied about the breakdown where you have acetyl-CoA being broken down and you get carbon dioxide and water and energy. Now we'll see how different products are formed from the intermediates of the pathway or cycle. So the first one, which is citrate, is known to form fatty acids. Citrate plays a major role in the transport of acetyl-CoA from the mitochondria into the cytoplasm for fatty acid synthesis. So remember, citrate is known to produce fatty acid. It is required for the formation of fatty acid. Next intermediate, which is alpha ketoglutarate, can form glutamate, which is an acidic amino acid, through the process of transamination or deamination. Glutamate in turn can form a neurotransmitter known as GABA. GABA stands for gamma amino butyric acid. It is an inhibitory neurotransmitter. Apart from that, alpha ketoglutarate can also form certain non-essential amino acids. Non-essential amino acids are those which are synthesized in our body and you do not require to take them through your diet. So alpha ketoglutarate can form non-essential amino acids. And these non-essential amino acids can be used in the synthesis of purines. Purines are nitrogenous bases such as you have adenine and guanine. The next one is succinyl-CoA. Succinyl-CoA can be used for the formation of heme. Okay, and heme is utilized mainly for hemoglobin formation and certain heme containing proteins. A few examples I could tell you is that of cytochrome, which is present in the ETC, electron transport chain. Then you have catalase, peroxidase, those are enzymes which are heme containing proteins. Then you have oxaloacetate, which can form aspartate. Aspartate is an acidic amino acid. Aspartate can be utilized for the formation of other non-essential amino acids, and those non-essential amino acids can also be utilized for the synthesis of furan and pyrimidine, again which are nitrogenous bases. Oxaloacetate can also be utilized for the formation of glucose through a process called as gluconeogenesis. Gluconeogenesis is a biochemical reaction where there is formation of new glucose from a non-carbohydrate source. So this is one of the reactions where oxaloacetate can undergo a series of reactions to form glucose, so that new glucose is formed into the blood, which can help maintain the blood glucose level, especially when the glycogen reserves of the body is depleted. So these are all the ways through which the intermediates can form various other important compounds in the body. Now what happens is, when you have all these intermediates forming various compounds in the body, over a period of time you will notice that the concentration of the intermediates within the TCA cycle tend to get depleted. So if it gets depleted, what happens is the TCA cycle will cease to function, and it may even stop. So that is not what is required, because there should be a constant energy production within the cell. So which is why there are certain reactions that can help to fill up the intermediates that are present in the TCA cycle. And that reaction is called as anaplerotic reaction. Anaplerotic means filling up reactions. Okay, so because of the depletion of the intermediates, you need reactions where you can fill up those intermediates within the TCA cycle so that the cycle can continue. So let's have a look at what are the different anaplerotic reactions. So we'll start off with alpha-ketoglutarate, which can be formed from glutamate again. The conversion between alpha-ketoglutarate and glutamate is a reversible reaction. So in either direction, the reactions can proceed. This happens through transamination or deamination. Then you have succinyl-CoA over here. Succinyl-CoA can be formed from the beta oxidation of odd chain fatty acid. Odd chain fatty acids undergo oxidation to give you a product called as propionyl-CoA. That's the end product at the end of odd chain fatty acid. So oxidation of odd chain fatty acid will give you propionyl-CoA, which in turn can get converted to succinyl-CoA. In addition, you have certain amino acids also which can form succinyl-CoA. For example, valine, isoleucine. Valine and isoleucine are branch chain amino acid. They contain branches in their structures. Then methionine, which is a sulfur containing amino acid. And even threonine, which is a hydroxy containing amino acid. All these amino acids can form succinyl-CoA. Then we have fumarate. You have all those aromatic amino acids which can form. Aromatic amino acids such as phenylalanine, tyrosine can form fumarate. And again, oxaloacetate. Sorry, I've left out malate. Malate can be formed from pyruvate through the help of the enzyme malic enzyme. Malic enzyme requires NADP, H and carbon dioxide. These are the two things required for the pathway to occur, the reaction to occur. The pyruvate gets converted to malate. So this is one instance where malate can be filled. And last but not the least, we have oxaloacetate, which can be filled up by the conversion of glutamate to oxaloacetate through the process of transamination. So this is the way. And yes, there's one more also. I missed out on one more. Oxaloacetate can also be formed by the conversion of pyruvate again, and this reaction is catalyzed by the enzyme pyruvate carboxylase, an important key enzyme in gluconeogenesis. So carbon dioxide is required, ATP and biotin. This is how all the all the intermediates are filled up by various reactions. Now, if you have a look at this, if you just see the entire citric acid cycle, it looks more like a traffic circle. Okay, where you might have noticed in a traffic circle where you have vehicles coming in and moving out of the circle. Likewise, we have the cycle over here, which is a circle, and you have various intermediates coming in and going out. Okay, so it's almost like a traffic circle. So with that, we are done with this session. To summarize what was done, definition, TCA cycle is the oxidation of acetyl-CoA to carbon dioxide and water. Energy is produced. Along with that, the intermediates of the cycle are utilized for the formation of important products in the body. Occurs within most of the cell, within the mitochondria, with the exception of RBCs which lack mitochondria. Then coming to reactions, there are 8 steps, 8 enzymes, and the total energetics formed at the end of the oxidation of acetyl-CoA is 10 ATP. Coming to regulation, there are 3 levels at which the cycle is regulated. That is at the level of citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. 2 other factors are also to be noted, that is the availability of substrates and the cellular need of energy by energy is also another criteria for regulating TCA cycle. Coming to inhibitors, there are 3 inhibitors. We have fluoroacetate, arsenite and malonate. The first 2 inhibits through non-competitive inhibition, whereas the last one is through competitive inhibition. And lastly, we have TCA cycle. It is known to be amphibolic because you have both production of important products or compounds formed in the body and breakdown of the major fuels, that is carbohydrate, protein and fat, into acetyl-CoA, which is ultimately used for energy production. So you have both catabolic and anabolic reactions occurring together. Hence, it is an amphibolic pathway. And because of the utilization of the intermediates for the production of important compounds, there is a need to fill up those intermediates so that the TCA cycle continues. So those reactions are called as anaplerotic reaction, which means filling up reactions. So with that, we are done with the topic of this session. See you in the next.